As early as January 2020, we were informed about this new pandemic. Initially, we became engaged above all at the local level in Italy to deal with the epidemic. But we immediately understood that the pandemic wasn't just a local Italian issue.
And the great fear was that if the virus were to take hold in countries where health resources were definitely at a much lower level than what we had in the West, that would have been a disaster.
The SARS-CoV-2 diagnostic project started in 2020 when the pandemic began and responds to a specific need of our developing countries, in particular in Africa, that had to become more expert in the diagnosis of SARS-CoV-2.
As part of our Council of Scientific Advisors, we have a Nobel Prize laureate, Sir Rich Roberts, who many years ago joined New England Biolabs, a company that over the years has become highly specialized in producing enzymes and had developed
a test that is particularly simple, fast, as well as inexpensive, which was a very good fit for environments with few resources, such as those in Africa.
We developed a test for COVID that is called RT-LAMP. We thought that this would be an ideal test for the developing world because it doesn't require sophisticated apparatus and it can be done at a single temperature and it can be done out in the field if necessary. And I should say that one of the key features is that it tests saliva, so it doesn't involve pushing sticks up into your nose.
So the first step was to test this method
in our BSL-3 facility, a high-security laboratory in which to handle these viruses that is also the only one available in the Friuli-Venezia Giulia region.
to then contact our collaborators in Nigeria, Cameroon, Kenya and Ethiopia.
Through our contacts, we were introduced to laboratories of excellence in their countries that had the ability to receive the technology and learn how to implement the tests.
We organised this clinical trial, comparing in parallel the new LAMP diagnostic test developed by New England Biolabs with the standard molecular swab test that is normally used for the diagnosis of SARS-CoV-2.
We all realized that this was a diagnostic that had to be established in a short time.
We remember a very difficult period from the point of view also of travel. What we normally do, that is, going on site to train the people involved in the study directly, was not possible.
Usually, when we start working on a project, we have to do studies for several years, deepen various aspects of the technology. At first, I was very skeptical that we would succeed in a short time to give a precise answer. Instead, after we did the first experiment, we realized that the system worked perfectly in diagnosing the presence of viral RNA inside the samples. In August 2020, we confirmed the validity of the test.
And the results of this study, on 1,500 patients, led to the conclusion that this newly conceived diagnostic test was almost as sensitive as the molecular test currently in use. It had sensitivity higher than 90% and was therefore able to identify the infection in the majority of patients.
And the results were much higher even than our expectations, because we saw that in those laboratories with enormous difficulties from the point of view even just to have electricity for all hours of the day, the kit worked in exactly the same way as it did in our laboratories in Trieste.
This is a classic North-South project that is a technology promoted in the north of the world that, through an international organization like ours, reaches what is called the Global South. And this is fully aligned with what the 2030 Agenda of the United Nations requires.
We have to remember that developing countries are often also the countries where pandemics begin, where there is the first passage from animals to humans. And so it is necessary not only to do diagnostics in the context of an already developed epidemic, but also to do surveillance, to identify new pathogens at the very moment that they begin to infect humans. And so all these tests must then be transferred to the member states, and the role of the ICGEB is precisely to facilitate the transfer from a molecular biology test and to make it sustainable and transferable to a country with characteristics that are less developed than those in Western countries.
It is also very important to recognise and remember the role of the Bill and Melinda Gates Foundation, who immediately believed in this project, both in the technology and in establishing this technology in Africa, and therefore funded the project.
With this project, truly, in ICGEB, we showed the best of ourselves, and we felt that our work did exactly what we were created for, and that is helping the developing countries.
It's a little bit like a snowball effect. You train one outstanding young scientist in Trieste, they go back to Nepal, and they can then subsequently train hundreds of scientists and young people back in the country.
Initially, I came to ICGEB because I'm studying DNA tumor viruses and the mechanism that causes cancer. I did my master's from Tribhuvan University, Nepal in biotechnology. Then I came here for my PhD in tumor virology. In Nepal was a huge earthquake. Obviously it was not a great time, but getting a news from fellowship from ICGEB was really like a dream come true.
ICGEB is an international organisation. That means it's made up of its member countries, not a single state. We have 66 members that comprise ICGEB. We have 3 research institutes which are located in Trieste in Italy, which is the headquarters, and also in New Delhi and in Cape Town. Overall, there's around 700 researchers working in the laboratories.
The ICGEB is a hub of fellows from all around the world and also a place with cutting-edge recent technologies.
We perform research related to many aspects of the SDGs: health, agriculture, sustainable energy, and so on and so forth.
The primary objective of my research is to study mechanism of carcinogenesis caused by human papillomaviruses. Today, more than 5% of all cancers are caused by human papillomaviruses. Even though a good vaccine is available, our therapeutic agent is limiting. One of the major standing questions in the field was to understand if the oncogenes that cause the cancer was important for carcinogenesis. I came to the lab tumor virology led by Lawrence Banks in ICGEB, where he was studying mechanisms of carcinogenesis. And using genome editing technologies, we could understand if expression of these oncogenes were indeed important for carcinogenesis. Carcinogenic mechanisms caused by these viruses is important for their therapeutic interventions, which would eventually save many lives.
We're not just doing research for research's sake. We're doing research which is relevant for our member countries. It's important to remember that the bulk of our membership come from the global south, or countries which will be considered as least developed or economies in transition. The fellowship opportunity and the training opportunities that they have in ICGEB are what I would call state of the art. It's world class. In Trieste, New Delhi, Cape Town, the fellows will come find an incredible international environment. There's people from all over the world. We are extraterritorial, and the facilities are really state of the art.
It was a perfect place with fellows from all around the world who are very friendly. The culture and place is really, really different from my country. The only thing I miss is good food back home with a lot of spices, but Italian food is also amazing.
For the fellowship program, the major aim of ICGEB is to provide training opportunities to young scientists coming from many diverse parts of the world. The aim being for them to be able to take those technologies back to their home countries and use that to further development of education and development within that country itself. What we want are young people who are incredibly motivated, have a passion for science, but also have a very strong desire to use science for the benefit of mankind. Sustainable Development Goals 4 and 5 are the cornerstone of ICGEB's fellowship program. We provide outstanding educational opportunities with obviously a major focus on ensuring gender equality, and we have developed a whole variety of state-of-the-art programs for providing access to quality education for women in many low-resource environments around the world.
I would really recommend fellows all around the world to apply to ICGEB fellowship and to learn.
In this time of pandemic, global insecurity, organisations like ICGEB are absolutely fundamental for bringing nations together and using science to promote cooperation and sustainable development.
The heart is an organ that is always in contraction. An abundance of mechanical stresses take place therein that in some way manage to reach the cells. The intuition of the ERASE project stems from two trivial observations. On the one hand, that the heart is an organ that does not know how to regenerate, and on the other hand, that it is an organ very rarely affected by tumours or from metastases. We asked ourselves whether applying the same mechanical forces exerted by a heart that contracts to an organ affected, for example, by a tumour, we can transmit the same signal to block the proliferation of growth. We realized that there were no tools to apply mechanical forces to the matrix in the cells, and therefore we had to invent these systems, thanks to collaboration with robotics experts.
I'm Domenico Pratichizzo. I'm Professor of Robotics and Haptics. I lead a research group that works between the University of Siena and the Italian Institute of Technology in Genoa. In the heart, tumours do not develop due to a mechanical phenomenon, that is, the heartbeat. We can use this knowledge also for non-cardiac tissues, using robots that can be worn and that induce on non-cardiac tissues, for example the skin, for example a melanoma, a mechanical stress very similar to a heartbeat and therefore have effects based on mechanobiology that slow the development of the melanoma and of the tumour.
As with every research project, ERASE was and is full of difficulties. We had difficulties understanding how to transmit these mechanical forces to cells, and to understand which forces are fundamental to apply. And then the greatest difficulty we are experiencing at the moment is to miniaturize the robots. Because when we want to work with cells, we have to miniaturize everything by a scale of at least 1 million times smaller.
These devices, we imagine, may weigh up to 100 grams, with a battery that can keep them functioning for up to 6 or 7 hours a day, whilst being almost imperceptible to the subject wearing it.
Another important challenge will be to understand which human tumours and specific medical conditions could benefit most from this type of therapy.
Today, we're ready with the technologies developed in my laboratory to attack external tumours.
For example, melanomas of the skin, rather than breast cancer, could be easier to attack with a mechanical therapy because they are external. We can then imagine perhaps wrapping these tumours in something that mimics a heart that contracts, to block their growth.
On internal organs, this is an even greater challenge, because we have to find technologies, mechanisms and robotic systems capable of transmitting these waves from a distance.
Fortunately, being able to collaborate with experts in the different fields has already allowed us to design small robots on which we have been able to cultivate cardiac cells and tumour cells. We see that, in fact, making these cells contract like a beating heart, they grow less. Thus, our initial hypotheses seem to have actually been verified. Hence, the challenge now will be to translate this hypothesis into something concrete that can actually block tumours.
Collaboration with ICGEB has been extremely synergistic. Our knowledge is strongly complementary. ICGEB is a very important centre from our point of view, because through engineering methodologies, it deals with issues that are important, including those related to biology and genetic engineering. And I believe that these are key for future technologies for the next 10 to 20 years. The ERASE project is centred on goal number 3, which is health and well-being, of Agenda 2030 of the United Nations.
Because, in fact, it proposes the development of new therapies, principally for tumours, with possible applications also in the cardiovascular field, and thus going to impact on those diseases that are the first cause of death in all the world.
Because it will develop highly innovative technologies for cancer treatment.
Tumours constitute one of the main health problems in the present society.
To date, 11% of deaths from cancer are due to lung cancer. For this reason, it is very important to study its development, and in our case, to study dendritic cells.
We deal specifically with a particular subtype of immune system cell called dendritic cells.
Dendritic cells belong to the immune system. They are called sentinel cells as they are very important in recognizing any pathogen or bacteria. For this reason, they are localized on the surfaces of organs that are in contact with the external surface.
Dendritic cells are important precisely because they are central to the capacity that our body has to respond to any kind of enemy, and they do so also in the context of tumours. These sentinel cells recognize the signals of diseased tissue and are able to alert the other components of the immune system and instruct them to start a specific and selective immune response for any type of tumour. For this reason, they are now frequently studied for their broad therapeutic potential to defeat disease cells.
Our purpose is to try to find therapies to prevent lung cancer, enhancing the role of these dendritic cells in the fight against cancer, and to try to strengthen them when diagnosis occurs, to activate them and to ensure that effective immunotherapy can commence.
In our laboratory, we deal in particular with how to ensure that the number and function of the dendritic cells in tissues remains optimal. This is because, if it is true that dendritic cells are potentially able to recognize a tumour, it is also true that the action of the tumour is one of suppression of the functionality of dendritic cells. They tend to disappear, to die, and therefore to reduce in number and efficiency. What we are doing is trying to identify molecules or treatments that allow us to keep their number always high, in order to maintain the maximum so-called immunological surveillance.
Dendritic cells in the lung are a very rare population, so being able to view them and identify them is very difficult. In the context of lung cancer, we are interested in studying dendritic cells using an instrument called a flow cytometer. This is used in the laboratory to separate and identify different populations from a complex tissue. In this way, we managed to obtain visual localization of both the dendritic cells, or healthy lung tissue, within the tumour, and identify the compartment around the dendritic cells through flow cytometry.
Immunotherapies are based on enhancing the immune response against tumours, but they still have limited effectiveness. In fact, the number of patients who respond is still restricted, and only some types of tumours respond to immunotherapy.
These are studies that require several years and therefore much commitment and perseverance to bring to fruition.
What we are doing will allow us, in enhancing the activity of this cellular subtype, to ultimately obtain benefit for an increased number of patients and for different tumour pathologies.
Furthermore, we must obtain funding
to update the technologies that allow us to keep up with the discoveries that take place throughout the world. This is a field in swift evolution.
Our studies in the fight against lung cancer fall completely within the context of SDG 3, the goal of Agenda 2030 of the United Nations that concerns health and well-being for all.
Our research represents an important challenge to improve human health and defeat serious diseases such as tumours.
Biopharmaceuticals have great benefits and today are much needed. Unfortunately, they are inaccessible to the majority of people on Earth. Access to medicine, to pharmaceutical products is a fundamental human right. Biopharmaceuticals are today the main players in the world of pharmaceuticals and one of the fastest growing segments of the pharma industry.
I'm Kevin Ainger. I'm Director of the Biotechnology Department at ICGEB. We are following a biotechnology program based on ICGEB's technologies for the production of insulin.
In recent decades, biopharmaceuticals have revolutionized the treatment of anemia, diabetes, autoimmune diseases, and tumours. They are recombinant proteins, such as vaccines and monoclonal antibodies, that are produced by living cells, for example in bacteria, yeasts, and cells in culture, thanks to the technology of recombinant DNA. The expiry of patents for biologics inaugurated a new era of products that are designed to be similar.
If you think it's just a copy, you may consider that there is not that much innovation in it. But if a drug's patent expired 20 years ago, the methods to develop it are too old, too costly, and not green. To make a copy, you have to invent everything from start to finish, and also take one step further.
The culture of animal cells normally grow in bioreactors. In this room, we inoculate bioreactors. With the help of this instrumentation, we follow the production of the monoclonal antibody or of the recombinant protein. Every step of the production process must be checked and followed. And here, every day, we control the cell growth in the bioreactors. The development of biosimilars has contributed to reduce costs.
The costs for patients are much less, by around 20 to 30%. This means that 3 times as many people can have the medicine.
And this has led to an increased usage, strongly driven by the greater availability of biosimilars. Thanks to the generous donation of the Friuli-Venezia Giulia region of €3 million, ICGEB has built new laboratories for the production and development of production processes for biosimilars. These are laboratories in an area of 215 square metres, composed of standard compliant clean rooms, in line with the standards of pharmaceutical industries. We will be able to develop technologies for the production of second-generation biosimilars that are recombinant monoclonal antibodies. The production process of the biopharmaceutical product ends here with quality control. In this way, our group also promotes innovation and capacity building at the regional, national and international level. ICGEB has signed over 70 agreements for technology transfer and quality control in the field of biosimilars, with 22 countries throughout the world.
About 3 years ago, we signed an agreement for technology transfer of insulin. Taking different technologies and transforming these from the level of advanced laboratory to industry. ICGEB helps many companies, or rather entire countries, to do this work.
We work to increase knowledge of pharmaceutical industries by supporting local production at a more affordable price. And these efforts work towards Goal 9 for Sustainable Development of the Agenda 2030 of the United Nations. Because all people should have access to medicines of good quality, and because access to medicine is a fundamental human right.
We have a worrying future in front of us. In 2050, there will be 10 billion people, and we will have to produce about 60% more food than at present.
Periods of severe drought, sudden temperature changes, an increasing number of people but less arable land.
As scientists, we must find technologies and ways to grow our cereals, our plants, also in marginal environments. Plants, like humans, are meta-organisms that live together with many microorganisms. The 2 millimeters of soil near the root, called the rhizosphere, is their intestine. In this zone, the plant exudes to recruit and maintain its microbiome.
That is, those microorganisms that help and increase the health of a plant, and that allow us to reach higher yields of food production. The microbiome of a plant is divided into different ecological niches. The microbiome in the root, the microbiome of the green part of the plant, that is, the leaves, the fruits, the stem, and the microbiome of the soil. That is precisely the reserve from which the plant recruits the good microorganisms, those that have a beneficial function for the plant itself.
Research into the microbiome exploded about 10 years ago. Beforehand, it was impossible even to think of undertaking this, because we knew that there were many very complex communities, but we couldn't study them. So what's happened in recent years? We've been able to sequence DNA very quickly, and it's very economical.
We study microbiomes through the use of DNA sequencing, and especially through the identification of gene markers that give us the name of the bacterium and that tell us which bacteria are present in a given situation and in what quantity.
With this information, we can understand who the main actors are, the bacteria that are at the heart of these communities, and that have the most important role in these communities.
Isolate them, characterize them, and use them in the form of bioinoculants to give to the plant.
There are also microorganisms that we can use to control diseases,
in order to replace chemical fertilizers that are still being used.
One of the methods we are studying together with companies is seed coating.
Coating seeds through the use of biofilms of bacteria.
Alternatively, these can be put directly in the soil before planting the seed.
The advantages of bioinoculants over chemical fertilizers are, above all, at the level of the environmental impact they have. We are not introducing any microorganism that was not already present in nature, but we are simply giving a greater bacterial charge of microorganisms to the plant that has a beneficial effect on the production and health of the plant itself. Moreover, the amount of available chemical fertilizers continues to decrease and costs more. Developing countries, for example those in Africa, have very little access to chemical fertilizers. Therefore, the use of organic fertilizers that will cost less than chemical fertilizers will be of great help.
The ICGEB is a unique organisation that has 3 research components.
The headquarters in Trieste and components in Cape Town and New Delhi. It's formed by 66 member countries.
We work together with these member countries. We have scientific networks because our mandate is
to promote science and research in developing countries.
Here at ICGEB, we have as our objective different scientific themes that align us perfectly with SDG 2 of the United Nations for Zero World Hunger, because we are studying many mechanisms that plants can use to be more resistant to pathogens and more capable of growing in extreme conditions. Different technologies, different scientists will lead to plant varieties that will be much stronger, and also to products based on microbiology that will allow us to increase sustainable agriculture in the future. So attaining 60% more food by 2050 for 10 million people is a very important challenge, and it's also worrying. But as a scientist, I'm very optimistic because I see many significant technologies, scientists who are very determined, and also scientific collaborations that will surely enable us to reach this great goal.
Defeating hunger in the world is possible thanks to the tools that we have, and the studies that we are bringing forward, thanks to which we will make land more productive and able to feed greater numbers of people.